I'll tell you what to do. Well, they probably won't be able to, because it's on the back of the clicker, right? But they so I'll say, if you lost your clicker, come down and break. Oh, you don't have to. Okay. I'm supposed to have a roll, so I'll have to sometime look it up on that, but I can't do it. Well, they were they from last week?
Yep.
Last Thursday?
Last Thursday they were here already.
Oh, okay. All right. Okay, I'm going to go ahead and get started on time for once, so let's go ahead and get in our seats. There are several announcements. Hi. Several announcements posted on Piazza. You need to go look at those. One of the big ones is the Quiz 3 was scheduled for a week from today. I just moved it to that Thursday, so I moved it from November 13th to November 15th, because I know a lot of you are still scrambling to get your initial contribution in. So you need to finish that up before the weekend starts, so you can put the weekend study for Quiz 3. So that's one big change. The other one is you do need to go before Thursday at about 11:30, and you need to download it and rank your group members, okay? And so I've explained how to do that. That's not a formal exercise. In other words, you don't get credit for doing it. There's a point assigned to it, and that's just for our record keeping, so that we can add up a column and see how many people have not turned that in. But you need to rank your group members, all of them, okay? And you don't rank yourself. And the spreadsheet is self-explanatory, has all the instructions. The way you name the file and return it to us is very specific, so be sure to follow those instructions. And so what you're ranking is the performance of your group members for Quiz 1 and Quiz 2. not for the posters, but for up to quiz 2, right? So that's like a midterm ranking. The final ranking will include performance, participation to the poster and all that stuff. That will go into the final ranking. Okay. Any questions about that? Yes?
Should we be concerned if we turned in exercise 3 that it hasn't been graded?
No. Those have to be individually opened and read, and there's a few hundred of you, so it takes a while. And on that topic, some of you are concerned, well, I didn't get to any of the talks. Well, that's kind of a problem. There will be more exercises. They're all going to be in the form of assessments, where those of you have multiple choice questions. And one of the things we're doing is we're skipping lecture 16. Lecture 16, because we're really running out of time. Lecture 16 is on the mitochondria. We skipped it last year. Probably one topic that you're most familiar with, you've started having since high school biology, is the mitochondria. What is the mitochondria? The powerhouses. Okay, so you know all about the mitochondria. So if you want to know more about the mitochondria, it's really important, okay? Go look at that. You should actually look at lecture 16 for 2 reasons, just to refresh your memory about what the mitochondria does. And second of all, I'm going to have an assessment over the mitochondria. So those of you that haven't been able to acquire points by going to lectures can do the mitochondria. If you've had biochemistry, you should be able to answer them straight away, okay? But if you can't, then you need to look at lecture 16, do the assessment, you get 5 points, okay? So there'll be some more of those. Yes?
How much do the lectures count for, how many points?
5.
5 per lecture? Or?
For those guest lectures?
No, like clicking in and stuff.
Oh, that doesn't count. No, we don't do it per point. It's just, you're either going to get 10 points or you're not.
Okay.
And that 10 points is part of the 50 participation. Yeah.
Okay, so you should be trying to go for 40 of your own.
Exactly.
Okay.
40 of your own. And once you get there, I mean, go ahead and do the exercises, but anything above a total of 50, in other words, you've been here every time, so that your attendance is going to be 10. Okay. Probably anything, a couple of absences is not going to affect. 3 absences becomes a real problem. Okay. But 40 points on your own is the maximum you can earn. So the total would be 50. That's the maximum. And everybody should get at least 50 points. But you got to earn it. Earn the points. Right? So I'm going to have exercises for you to do. And there may be 1 or 2 more lectures you can go to. So
If you get over that?
it doesn't do anything except edify you. Okay.
So the 10 points, you should get it unless you miss a significant amount of lectures.
Exactly. Yeah. Okay. Other questions? Okay. Let's get to, we're going to finish up trafficking today, I hope. And that will be the end of quiz 3 material. Okay. Just because I'm moving it, I'm not going to add extra lecture material. So this will be today's lecture. I'm pretty sure we'll be able to get to the end of it. Okay. So finish trafficking. And so we're talking about an organelle here it's fairly complicated. That is the Golgi apparatus. And the Golgi, remember, one of the characteristics is each cisterna has a particular function, okay? And there's no need to memorize all this stuff, but there's different enzymes and different Golgi stacks to do different things, okay? And we'll look at a couple of specific examples. Those are the only poster children you're going to need to know about which enzymes do what, okay? And how do each of these stacks maintain their unique composition? Well, we talked about that last time. There's a couple of different models. There's the vesicular transport model, which we don't, there's fewer people that ascribe to that model than the cisternal maturation model. What's the key feature? What's going on here? One of the key features, differences between this and the vesicular transport model is that actually what forms the vesicular tubular clusters are vesicles that are coming off the ER, and they undergo homotypic fusion, right? And so in this model, the vesicular tubular cluster becomes the cis-Golgi network, okay? It actually matures into and becomes this. Then each of these cisterna move forward So it's cisternal maturation. They're moving forward on a series of railroad tracks, which are microtubules that underlie these different stacks. Okay, so the stacks are moving forward, and the way you maintain the unique composition is to send vesicles retrograde. Okay, and a specific type of retrograde vesicle. It's COP1 coated. Okay, now that violates. That's typically what you use to send traffic back to the ER, so that makes a lot of sense, right? So COP1s are sending enzymes to the previous cisterna to maintain its identity as this thing continually matures and moves forward, okay? And then what happens at this level is what's going on is that's where a divergence of different trafficking sends stuff to the lysosome, to the plasma membrane, and to regulated secretory vesicles. Okay, so now, so let's talk about lysosomes. Lysosomes are probably more interesting than what you think. Lysosomes are heterogeneous. The hallmark is that they're very acidic, and we have a couple of different pathways to get cargoes to the lysosome. One of the pathways, so there's 2 synthetic pathways and 3 digestive pathways. As you probably remember from freshman biology, the lysosome is sort of the trash can of the cell. It's very important because if you don't chew up old components, they remain in the cytoplasm and take up space as inclusion bodies. And so you don't want to have a bunch of junk taking up space in this very useful cytoplasmic compartment. So what happens is lysosomes, if they're acidic, you have to have a way of maintaining that acidity. And so what you're going to do is you're going to target the VOV1 pump to the lysosome. And it's the VOV1 pump that's creating the pH 5 that's necessary for these enzymes to work. One of the safety mechanisms you have, that the cell has in case this ruptures, is that these enzymes don't work at physiological pH of the cytoplasm. So even if this ruptures, these enzymes aren't going to go around chewing up stuff. If these enzymes are delivered to the outside of the cell, what's that pH? What's the pH of your blood? 7.2, 7.4, something like that. These aren't going to work, right? So this is not going to chew yourself up if there's an accident. And accidents do happen, okay? So these things work at only a pH optimum of 5, very acidic. That's created by this proton pump. So there's tons and tons of enzymes to destroy everything it's going to encounter. Nucleases, proteases, blah, blah, blah. Now, there's 2 types of enzymes. There are soluble enzymes, okay? And then there are membrane-bound enzymes and receptors. And so those two get to those 2 components reach the lysosome by different pathways. Here's the rule. If you are a membrane-bound enzyme or cargo receptor, the way you get to the lysosome is you go from the Golgi, you butt off into clathrin-coated vesicles, okay? And what's mediating that clathrin-coated vesicular traffic is not AP2, it's AP1, okay? AP1. And so you go straight to the late endosome, and here's the rule. Anything that goes to the late endosome is going to go to the lysosome. In fact, you can think of the late endosome as a different maturational state of the lysosome. It is the lysosome, where it's going to mature into that. So reaching the late endosome means you go to the lysosomes. All right, so that's how you get there from the Golgi. Now, if you are a soluble enzyme, you actually have to go from the Golgi. Again, you butt off in clathrin coating, same kind of clathrin coating, AP1 mediated clathrin coated exocytosis, and it goes to the early endosome, and then it goes to the late endosome, and then to the lysosome. So one question you should have, why go to all that trouble? It seems like you could spin off and you can miss your target, and sometimes that does happen. Things end up in the plasma membrane instead of where they're supposed to go. So there's a reason why we go this way for soluble enzymes, okay? So those are the 2 synthetic pathways that construct the lysosome. And those are listed here, of course, okay? Now, there are 3 different digestive pathways, because this is the main function of a lysosome, okay? And so this is just pointing out that the late endosome becomes the lysosome. So things that are going to be digested have to fuse first with the late endosome. and then that matures into the lysosome. Okay? So let's talk about these digestive pathways and when we use them. Okay? So we have three different types of traffic. They're going to fuse with the lysosome. You have endocytotic vesicles that come from the plasma membrane, and we're going to talk extensively about the early endosome. So the traffic coming from endocytosis is going to fuse with the early endosome. And if you're destined for destruction in the lysosome, you have to go from the early endosome to the late endosome. So that's the rule. To get to the lysosome, you got to go through the late endosome. Okay? So if you fuse there, you are what? You're committed to go to the lysosome. If you end up in the early endosome, there are some other choices, though, okay, that can be made to rescue you before you hit the lysosome. So this is one type of digestive traffic. The other type are getting big things to the lysosome for destruction. Let's say you're a macrophage, and so you ingest a bacterium. That occurs by a fundamentally different type of endocytosis. It's known as phagocytosis. And we'll talk about the distinction, but basically you actually pinch off this huge phagosome. How does the phagosome deliver its quarry to the lysosome? It must fuse with the late endosome. That's the rule. Okay? And then it becomes the lysosome and it's digested. Now, this is a fairly newly described pathway. This is autophagy. If you want to be really snippy about it, you call it autophagy. Anyway, I like autophagy. It helps you spell it better. Okay. Now, autophagy is constructed slightly differently because basically we're not sure where this membrane sac comes from, but we think it's a tubule from the ER. And the ER tubule forms a double membrane barrier around whatever that needs to be destroyed. Your mitochondria wear out, okay? They get lots of oxidative damage, as some of you know, because you're doing projects on that. And that mitochondria needs to be gotten rid of. Or it could be some other old piece that needs to be digested. So let's take an example of the mitochondria, okay? And so you wrap this tubule around it. So this is like the nuclear envelope. It's a double membrane, isn't it? Okay? And so once you do that, you form what's called an autophagosome. And the only way you can destroy it is to do what? If you're going to go to the lysosome, where do you have to go first? Late endosome. So everything flows into this pathway, and then it becomes the lysosome, and it's destroyed. And what happens is the cell reclaims all those nice nutrients that constructed the mitochondria. Okay, let's do a problem to test whether you understand the difference between this pathway and that pathway. So if let's pretend that you're an oxygen molecule, okay, and you're outside the cell, and you're going to diffuse into this mitochondria, or you're going to diffuse into that mitochondria. Let's pretend that's a mitochondria up there, okay? Let's say that is right up there. Let's get rid of the bacterium. And a mitochondria really is a bacterium, isn't it? That's where they evolved from. Okay, so how many lipid bilayers does that oxygen molecule have to cross from the outside of the cell to get into the matrix of the mitochondria? What's oxygen do in the matrix of the mitochondria? What's its role? Why do we breathe oxygen? Final acceptor of electrons, okay? It has to, right? And it turns into a water molecule, right? Okay? And so we know we need it, so how many lipid bilayers does it have to cross if it goes from here into there? And how many bilayers does it have to cross to go from here into there, if that's a mitochondria? It's outside the cell. I'm sorry. Into the matrix of the mitochondria. Okay, go ahead and work that out. Everybody has to think about this. Let's focus just on this one. Tell me how many, and what we can turn this into a clicker question. So I'll put this up here. If you think it's 1, click A. If you think it's 2, click B. If you take it's 3, click C, 4. Of course, you can't click more than, it could be more than 5, but let's say 5 is the limit. Okay, just a few more seconds. Go ahead and click so I know you're here today, even if you don't get it. Okay. Is that it? Okay. Okay, answer 4. So I guess it only took 4 answers. Well, that's going to be bad because that doesn't allow you to answer the correct answer, which is 5. That's a trick question. I won't ever do that on an exam. Okay, so it has to diffuse through one lipid bilayer. That's the plasma membrane. It's got to go through that lipid bilayer. That's number 2. It goes into the lumen here. Then it crosses the second. That's 3. Okay, then it's got to cross the outer mitochondrial membrane. That's a lipid bilayer. And then it's got to cross the inner mitochondrial membrane, which is 5. Okay, so there's 5. Now, is that different from this? Yeah, it's got to cross 1, okay, to get across the plasma membrane, and then it's going to, this is a single membrane, so that's 2, and then 3 and 4 from the outer and inner. So it is, you actually have to cross more lipid bilayers to get into the autophagosome than you do to get into the phagosome that's got a mitochondria. Yes?
Would you ever really save oxygen to a mitochondria that's not being destroyed?
It's still going to take it up, isn't it? So it doesn't know if it's good, bad, or ugly. And so it would still go in there. But this is just a question. But when does this actually come into play clinically? Well, it actually becomes very important because it turns out that those stupid cancer cells that we all talked about at that, how many people went to the thing? It was a pretty good showing. Okay, pretty good talk. Very interesting about approach to cancer. What happens is the cancer cell tries to evade destruction by any means possible. Even if you, so if you give this the tumor a anti-angiogenic factor, which prevents that tumor from supplying itself with blood vessels. Tumors actually release these angiogenic factors. Blood vessels grow into it, and then the tumor can get bigger and also can spit off metastases to elsewhere in the body because it actually vascularizes itself. So one cancer treatment is to cut off the blood supply, right? Okay, well, the tumor could be much sneakier than you think it is. What it does is it's not getting very much oxygen, it's not getting glucose, so it starts to eat itself. Actually, tumors survive through undergoing autophagy or autophagy and stay alive that way. That's an escape pathway that tumors, they're sneaky. I mean, they will try anything to survive. Okay? So this process is actually covered probably from a lot of people that are looking at tumor biology. Okay. All right, so, we got to go to this. It's always difficult. Okay. So we need to talk about the why is it that the pH is so important? We know one reason why the pH needs to be low in lysosomes because that's the safety mechanism to destroy the stuff that you ingest and send to the lysosome. But the other compartments are also acidic, and so there's different roles for that acidity. Okay, so let's talk about this pathway. This is the pathway to get lysosomal soluble enzymes into the lysosome. So this looks pretty complicated, but it's actually even more complicated than that. We have to put a tag on lysosomal enzymes that are soluble to get them to go through this sort of circuitous route, okay? And how do you do that? So let's look at this process. So this is the tag that has to be added to a lysosomal hydrolase that's soluble, okay? And it is an N-glycosylation. So if you're a lysosomal enzyme, you must be N-glycosylated, okay? And the reason that that is true is that if you're N-glycosylated, you will end up having a terminal mannose, okay? Because remember, the hallmark of N-glycosylation is high mannose number, okay? And so you'll get this mannose sugar on the outside of this little oligosaccharide tree, okay? And what we're going to do is we're going to phosphorylate the sugar in the 6th position. So what we create is what's called a mannose 6-phosphate tag. And that's the zip code to go to the lysosome and become a soluble enzyme. Okay? Now, how do we construct this tag? Okay? So the protein has some information in it that I'm a lysosomal enzyme. So there's a special patch that identifies it as a lysosomal enzyme, but it also has to have this tag. So we need to actually have a binding site for both of these things. So where does the tag get added? It actually is a little complicated. The first place that it happens is in the cis-Golgi network. Okay, so that's the very first stack that is formed in the cis-Golgi. Okay, and there's a binding site that recognizes the enzyme, and then there's a binding site that is actually enzymatic in nature, and it's actually going to take UDP N-acetylglucosamine, which is a mouthful. Okay, it's just a sugar carrier, and it's going to put that sugar onto that 6th position, okay? It's going to link it with a phosphate group, but then it's got this weird N-acetylglucosamine little sugar group, okay? And if you don't remember what N-acetylglucosamine looks like, you can go look it up in chapter 2. It's irrelevant, okay? So it's got the signal that we're looking for is mannose 6-phosphate, but it's still masked. It's a masked zip code. It's like hiding the zip code as long as we're in the cis-Golgi network. So what happens is it goes through the cisternae, okay, how does it get there? The cisternae mature, they move forward, and when it reaches the trans-cisterna, not the trans-Golgi network, the trans-Golgi cisterna, that's where that little tag is unmasked. So what we do is we clip off that little N-acetylglucosamine in the trans-Golgi cisterna. Clip it off. That reveals its true nature, which now it's a mannose 6-phosphate tag. And when it gets to the trans-Golgi network, that's where it's going to send it on to its next destination. So it's kind of a complicated process of putting these tags on here. Now, what happens is there's some people that have a genetic disease called ICEL disease. And what happens is they don't have this enzyme. So they can't put the tag on their lysosomal soluble enzymes. So where are they going to go? What's the rule? This protein got into the ER as a secretory pathway because it had what? If it's a soluble enzyme and you're in the ER lumen, how did you get into the ER lumen? There's only one way to do that. I mean, there's one major way. There's some weird ways to do it. And where's the signal peptide located? At the N-terminal. But that's no longer here, right? It got cleaved, and so you're a soluble enzyme. You need to go to the lysosome, but you can't put the tag on. So where are you going to go? What's the default pathway? Out of the cell, into extracellular space. So those people, what happens is, it's only a certain type of cell, particularly, I forget what the cell type is now. Fibroblast, okay? And so these fibroblasts, what happens is they can't digest all these things, and so they end up with fibroblasts that are chock full of inclusions. And so that compromises the function of the fibroblast, and as you know, fibroblasts are very important. And so that's why they call it ICEL disease, inclusion body disease. Okay? What's really weird, though, is in the liver, their lysosomes work just fine. So that must mean there's another pathway of tagging and and directing traffic to the lysosome, but we won't talk about that. But this is the major pathway to get something to go to the trans-Golgi network. Now, how does it get it to go to the lysosome? We still haven't answered that question. Okay, so we're going to get it to the trans-Golgi network. This is where we unmask the tag in the trans-cisterna. Then it goes to, what's the function of the trans-Golgi network? That's the sorting station. That's Grand Central Station, which is probably flooded right now. And so we got to go this way. We don't want to go this way. We don't want to go that way. We want to go this way. So how do we do that? Well, we put that nice tag on there, so we've got a zip code. And so in the trans-Golgi network, there's a receptor that's going to recognize that mannose-6-phosphate. Guess what it's called? The mannose-6-phosphate receptor, okay? And so that mannose-6-phosphate receptor, let's look at that. I've shown you this picture before. Now you know what's going on here. The mannose-6-phosphate receptor is a transmembrane. It's got one alpha helix, okay? And so this is the side of the protein that's recognized. See, this is the trans-Golgi network. It would recognize the cargo, which is the lysosomal enzyme with that mannose-6-phosphate tag. So that's why that receptor binds to it. And then how does it know to bud off into a clathrin-coated vesicle? It does because ARF comes down, ARF splits or loses GDP, it picks up GTP, it becomes active, it recruits, Nikki up here, and this is AP1 instead of AP2, because we're at the trans-Golgi network, not at the plasma membrane. As you know, AP1 is going to recruit the cargo receptor and also the clathrin. Okay, so it's going to do all that good stuff. And this will bud off into a clathrin-coated vesicle. And where's it going to go? Well, what happens is, it's not the typical thing. What happens, it doesn't go directly to the late endosome. What it does is it goes to the early endosome. Because what's going on in the early endosome is, it's not acidic enough to activate the enzyme, but it's still about pH 6. So it's still pretty acidic. And what that does is it releases the cargo from the mannose-6-phosphate receptor. The cargo receptor dissociates under the influence of the low pH, okay, the high proton concentration. So the cargo is now soluble like it's supposed to be. Then another enzyme is going to clip off the phosphate. Why would you want to clip off that phosphate off the mannose moiety? What's that going to ensure? Why would you want to do that? You're getting, what you're effectively doing is destroying the zip code. Well, what you want to do is you want to send the mannose-6-phosphate, that's a lot of protein. You want to send the mannose-6-phosphate receptor and recycle it back to the trans-Golgi network to pick up another round, right? So you don't want the receptor to go to the lysosome. So it's going to be shipped back to, recycled back to the trans-Golgi network without the cargo. So you don't want the cargo to rebind to this and get shipped back to the trans-Golgi. That's probably not going to happen because this is acidic enough. But what if the VOV1 pump's not working very well that day? And this doesn't dissociate, well, that will just ship it right back to the trans-Golgi network. But if you cut off that phosphate, it won't go back. Okay? And so this is then going to be budded off into a membrane process that we won't talk about. It goes from the early endosome to the late endosome, and then the late endosome means it's going to a lysosome. Okay? So that's how you get a protein into the ER all the way through its tagging process. It's masked, it's unmasked. It then goes to the... So the reason you go through that circuitous pathway, let's redefine it. So this is the pathway. You go from here to there, and then there. The reason you go to this is to uncouple the receptor and reuse it. Okay? That's the reason. It saves energy. Okay. Any questions about how that works? Okay. And so why don't you do that for enzymes that are membrane proteins? I'm sorry?
You wouldn't need a receptor for it, would you? Because it's showing its face to the cytoplasm.
Right. So it can bud off. So an enzyme that's a single pass membrane protein can be shipped directly to the lysosome because you don't need to, there's no cargo receptor to recycle. It is the cargo receptor. It is the cargo. So you don't need that. So you can go straight into the lysosome, straight into the late endosome, right into the lysosome. Okay, so where does that leave us? So we've constructed this tag, and then I went through some of the real basics of endocytosis. Your book considers all cellular uptake endocytosis. Now, there's a lot of cell books that distinguish between endocytosis and phagocytosis. Your book thinks that phagocytosis is a type of endocytosis, so we're going to go with that, right? So there's two types of endocytosis. There's phagocytosis, and then there's cell drinking, panocytosis. What we've been talking about with clathrin-coated vesicles, bringing transferrin into the cell, that was our poster child when we first talked about AP and ARF and all that stuff. That's panocytosis, and it's specifically called clathrin-mediated endocytosis. We used to call it receptor-mediated endocytosis, but those terms are interchangeable. So where this receptor-mediated endocytosis is a very specific form of panocytosis, whereas the cells actually are always drinking and eating, okay? And so some of that is just constitutive, and that's not clathrin-coated. It's clathrin-independent. That makes it constitutive. But this is very specific. Like, I want to take up the transferrin molecule, so I need a transferrin receptor. That's why you use this. Okay, we'll talk a little bit about here in a moment why phagocytosis is very different from this type of clathrin-mediated endocytosis. Okay, we can actually skip this because that's just a rewrite of stuff that we've already talked about. What I was focusing here on and what your book focuses on is a very specific cargo. Let's look at this. This is clathrin-coated process coming off the plasma membrane or coming off where? Trans-Golgi network. They're both clathrin-mediated, okay? And you can tell a clathrin-coated vesicle because it looks like a little soccer ball. But understand, we don't want to be naive and think that that's the only type of vesicular coating. There is a different type that is mediated by caveoli, these little pockets here, and we're not going to talk about those. And they look a little bit different. They almost look like a cabbage is what it looks like, that type of out pocket. But there's very specific information about what goes into those types of coating. We're going to stick with clathrin as sort of our poster child for endocytosis, receptor-mediated endocytosis. We're not going to worry about the caveoli. Okay. Now, medically important is this example. All of you probably have heard of LDL, low-density lipoproteins particles. Okay. And so what makes an LDL particle is the fact that it has this protein on the outside of it. It's a apolipoprotein like this, and this is what allows this particle to be recognized by the LDL receptor. It doesn't recognize the whole particle. It binds to this very specific protein that's wrapped around here. Now, why would you want to take up LDL particles? What's in here? Lots of cholesterol. Okay. What type of cell uses lots of cholesterol? What fun things can you do with cholesterol? Steroids. You can make steroids. Okay, so the ovary, the testes, the adrenal gland are all going to have lots of LDL receptors and take this up, right? And that's simultaneously, that's going to get this bad cholesterol out of your circulation. The people that can't take this up actually have a familial form of hypercholesterolemia. It means they have huge levels of cholesterol in their blood. And the reason that we're going to actually, I'll show you exactly where the mutation is, okay? So let's take up that cholesterol, and we know how this happens. You can transfer what your knowledge that you know about the transferrin receptor just to here, okay? And so we switch out receptors. We have an LDL receptor. Again, it's a single pass membrane protein. What's going to happen is it's going to bind its cargo, and then the ARF is going to bind. It's going to pick up GTP and lose GDP, It's going to stick to this membrane, right? The cytoplasmic side of the membrane, and it's going to recruit AP2, and AP2 is going to recruit the clathrin coat, and this thing will butt off into a clathrin-coated vesicle. So we don't need to go through those details. But what's critical here is this little cytoplasmic domain. Okay, if you're an LDL receptor, you have to have a very specific sequence projecting in the cytoplasm. There it is, right there. Acid pro-val tyrosine. And people that have familial hypercholesterolemia have a mutation in this little consistent sequence. And so the LDL receptor is not recognized even though it's binding to the LDL particle. And the LDL particle just sits there and dissociates. Okay? It stays in the blood, and then these people end up getting heart disease, usually have a heart attack before age 20. Okay. And so they have to take all kinds of drugs to lower their cholesterol levels. Okay. So this is medically important. And I'm sorry? It's a genetic disorder. No, it's not affected by how you eat. Okay. All right. You know they must be on a very, very low cholesterol diet to survive. Okay, so now, we only know part of the story, right? Where does the LDL particle go? You're going to need to digest it, and that happens in our friend, the lysosome. So what happens is this gets taken in. The first thing that happens before it's confused with the early endosome is uncoating, and you know how that happens. Go review it in your notes, okay? So this is now displaying the targeting and fusion information. It's got the RABs and the tethers and all that stuff, right? So it's going to fuse with the early endosome. Why is it going to do that? It's going to do that for the same reason that the mannose-6-phosphate receptor fused with it, which was, why do you go to the early endosome instead of straight to the lysosome? To recycle the receptors and reuse them. So we're going to come into this compartment and uncouple the receptor from the ligand. In fact, this used to be called the curl, compartment of uncoupling a receptor from ligand, until people started making fun of that name, the Committee for Unscientific Replacement Letters. So they got rid of that name, okay, and then so they named this the early endosome, right? So it shoots off transport vesicles filled with what? Just the receptor, because remember, this is at pH 6. And so the LDL particle is actually going to uncouple from the receptor and go back into a transport vesicle, and then it goes to what's called the recycling endosome, which sends all of its traffic back to the plasma membrane, and this arrives back here and can pick up a new round of cargo. Right? So we've saved biosynthetic energy. Meanwhile, we use a different type of vesicle budding to send this to the this would go to where first? Late endosome, and then it becomes a lysosome and destroys all that nice, breaks it apart, gets rid of the protein, reclaims the amino acids from the LDL particle, and recruits all that cholesterol and uses it to do what? To make steroid hormones and to put it into the plasma membrane. Okay. So, it's the change in pH is what uncouples this. We actually know why that change in pH uncouples the receptor from the LDL particle. Okay, so let's take a brief look at that process. So you have, this is the early endosome. Is that the focus? Yeah, okay. And what happens is, here's the LDL particle. This is the LDL receptor. And it has a domain that's actually, it's got a what's called a beta propeller motif. Because it's got with this beta sheet, it looks like a little spinning propeller. Okay. And that is right there. Okay. This is actually it has a little, what's called a ligand binding arm like this that is negatively charged. Okay? So it's got negatively charged amino acids. And this LDL particle has an affinity for those negative charges. Okay? So as long as the pH is neutral, this will hold on to that, right? But now we're in the early endosome. What's the pH? The pH is now dropped to 6.0 because of the VOD1 pumps. Okay. So at the plasma membrane, this receptor works really good because at neutral pH, this little arm picks up the cargo. Okay. But what happens in low pH? What happens is the face of the beta propeller motif starts to pick up positive charges. All those at high concentration protons protonates the face of that beta propeller. And what that does is it attracts that ligand binding arm, it pulls it into the face, so that will then become something like, if we drew another one like this, that face is now positively charged, and this ligand binding arm is now attracted to the beta propeller. And that releases the cargo, the LDL particle, into the early endosome fluid, where it can be taken up in a transport vesicle and sent to the late endosome. Where is this guy going to go? We're in the early endosome. Where is it going to go next? To go back to the cell surface. What are we doing to this receptor? We are recycling, so it's going to go to the recycling endosome, and then it goes back to the cell surface. But this is, at a very biophysical level, this is how you uncouple the receptor from the ligand. Okay. Any questions about that? Good. This is just, it's hard for me to draw a three-dimensional, it looks like a cup. That's one symbol. Right. You don't need, there's two pieces you need to know. The beta propeller motif, which is this little blob here, and then that little ligand binding arm, those are the two pieces that change charge. Okay. Let's take a break there. Just leave it. Yeah.
Two questions. So this transformation is early in the summer because the VOD1 pump was at 6, VOD1 pump kicks in,
It's already done its job. It's already kicked in. In order to create that early, I should be showing this inside the transport vesicle that's coming from the cell surface. Because at that point, it looks like that. Okay, so what's going to happen is at pH 6, it becomes like that. I'll reestablish
Well,
that.
traveling needs more base, pH 7 or so.
Right.
And as it's transported to 6, you get this more acidic. Second question is, so I went to the podiatry. I talked with a lot about it. I'll ask this one.
Oh, that's okay. 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 Did you email me? Oh, you did. Okay. Somebody else. Okay. Yeah, let's just set up a appointment. Yes. What's a good time? Not in office hours. What's another time that you? So tomorrow afternoon. Let's see. What do I have tomorrow afternoon? I have the graduates. We meet at 2:30.
It'll be 2:30 to like 3.
Because we only have one paper. I need to also mention that. So, 3:30? Would you be available at 3:30? So just come to my office at 3:30. But send me your name, so I'll put your name. I like to put your name on my calendar. Yeah, and send it to Blackboard. Okay.
So you didn't quite address it, but that it leads to it. When you was talking about the carpet bomb, how like we had developed the carpet bomb, and basically that's what happens with the projects. On the review of the treatment, you have that concept, but you also have how drug treatment causes the bottleneck, and you have that window right after the drug treatment to hit. You have this heterogeneity, and you bottleneck it, and you have a homogeneous because you have few projects. Right.
It's abstract.
So
to turn
The
in.
timing of the treatment. You can't just give it all at once.
Exactly.
First of all, you kill the patient.
Yeah, and you have systemic damage at a distance.
Right. He didn't talk about it, but the reason those inhibitors of the pumps, the reason they don't work is that the therapeutic dose is very close to the lead.
Yeah,
And so you can't.
and that's in my research is on that stuff. So that's why damage it linked for me. My question to you is: do you think that obviously it is a combination, but what do you think is on the forefront? Do you think it's the carpet bomb of the heterogeneous subclans, or do you think it's targeting the homogenetic body?
Well, I think targeting is
Targeting in general.
and I was going to ask him because he's more up on the clinical side of it. Has there ever been any nanoparticle that's been fit with targeting antibodies and weaponized drugs? Has any of those particles have they worked? Have they gone just to the tumor and not made the patient sick? So
And see, like I talked with Dr. Butler, his concept was, well, in the next 5 years, we need to beef up natural killer cells to hit those projects.
yeah, we could use our own
And then,
and come up with a vaccine.
in the next 10 years, we could have this NK subclonal expansion to target the cleaner. So it's pretty cool.
It is.
I just wanted to get your opinion.
I'm just scared of messing with the immune system because that's how you get lymphoma to begin with. So,
Dr. Butler's pretty confident.
well, that's just his thing.
I was curious with the late endosome. I know you said like it matures to the lysosome. Now, does it fuse with the lysosome, or the vesicle? So the vessels are delivered to the late endosome to make it a lysosome, but it actually fuses.
It actually.
The individual projects. Yeah.
For the, maybe like for books that are just like online.
I would go for what you want is Okay, why don't we go ahead and get started? Okay, graduate students, you probably know that we meet tomorrow for just one paper. If you've already sent your questions in from last week, you don't need to resend them. Okay. One other clarification about this. This is confusing. This really should be called a transport vesicle that's going to fuse with the early endosome. This is what happens in the early endosome. This is what would be happening if this is coming off the plasma membrane. Okay, so this would be an endosome, right? And at that point, the pH is not 6 yet, because if it's 6, this is going to be dissociated from that. So this would be less. This would be a higher pH, more alkaline. Okay, and that's why this little ligand binding arm is still attached to the LDL. When it fuses with the early endosome, which you should take that and put it down here, what happens is the pH is now 6. That the beta propeller surface is now positively charged because of the protons, and then it's going to pull that ligand binding arm and retract it, and that releases the LDL part. Okay, I didn't mean to confuse people. Okay, let's take that off and shut it off. Okay, so let's do a couple of problems here to get back into the swing of things. Oops, let's go back. Stop this. Okay, and we're not going to do this problem. We have to do. Okay, I realize this is a very wordy problem, and actually you're not going to be able to because this is sort of a what's wrong with this type of question. So ignore we won't do the clicker thing. You couldn't have possibly answered, so that's kind of ridiculous. All right, so okay. What's wrong? The question here is what's wrong with this description? Okay, now talk to your group and see if you can come up with a central problem with this description. And that means you should actually speak to them in English. Or Japanese, whatever your favorite language. Okay, who has group? Group 10. Oh, you want to. Okay, where's group 10? Group 10? Nobody's here for group 10? Nobody wants to admit. Are you guys group 10? Group 10 has 5 seconds to identify yourself or you're going to lose 10 points. Yay! Okay. Nobody wants to lose participation points. Okay, so is there any problem? Maybe this is completely accurate, right? Where does it stop? So you got a cargo and you want to take it from where? ER to the Golgi. And so do you need a gaff? Yeah, you need a gaff, right? And so that's going to help impose directionality, right? You're going to pick up you're going to activate SAR at the ER, and that's going to help recruit the COP. Is it COP2? Is that correct? Yeah. It's going anterograde from the ER to the Golgi. It's COP2, right? This is correct. And SAR has to be in the GTP state in order to bind to the ER and recruit what? What does SAR recruit? AP1? No, wrong system. Right? It recruits codamers. It's what kind of codamers? What kind of protein? Give me a hint. Starts with a sec. Nope. First thing is 23, 24. Okay. Now, okay, what this thing says then the gap is located on the Golgi membrane. So somehow you're going to coat this, uncoat it, go to the gap, and the gap is going to. Yeah, so it starts to be really confusing right here, right? So this is what's wrong. Okay. So what's wrong about it? Where is the gap for SAR1? It's on the ER membrane, right? So it's pulled, so it's actually part of the COP2 coding mechanism, and it is specifically SEC 2324 is the gap. But remember, SEC 2324 has to recruit an outer code, which is SEC 1331, and SEC 13. So when this thing buds off now, the way it uncoats is SEC 1331 activates SEC 2324, and SEC 2324 is the gap, and then it is what it causes SAR1 to split the terminal phosphate and send us back to the GDP state, so it retracts its landing gear, and then causes the whole thing to fall apart. Right? So very specific about how this works. You need to know all those steps, okay? Let's do another one real briefly. Okay, and this one we can do with the clickers. So try this by yourself. This should be a vocabulary. Okay, a few seconds, make sure you're here today. There is a sign-up sheet also, if you can bring a clicker. Okay? All right, answer 3, which would be, let's get rid of this now, would be AP2, that's correct. Okay, and so now we have a little bit more sophisticated. AP2 is determining trafficking coming from plasma membrane. AP1 is from the trans-Golgi network outward. Okay, so AP1 and AP2 are different. Let's get rid of that. So we need to be able to, just got a couple of things that we need to do. Contrast endocytosis and phagocytosis. That's actually very, fairly simple to do. We'll get to that in just a moment. What we need to do is finish up this little piece about endocytosis, receptor-mediated. There's 3 different types of fates for materials that come into the cell by receptor-mediated endocytosis. One is, I think we already talked about LDL particles, okay? Pulling in those, recycling the cargo. You have specific proteins, certain cargo receptors, but not all were retrieved from the early endosome and recycled. That's the LDL. When do you recycle the receptor and when do you not? Okay, so if you're going to need a lot of stuff, that means it's sort of a constitutive thing. You need lots of transferrin, you need an iron, you need lots of LDL. Those things are going to be recycled because you want to use the protein over and over. When is it that you actually want to send the cargo and the receptor to the lysosome for destruction? Well, you want to do that if you're a dangerous receptor, okay? And a good example of a dangerous receptor is epidermal growth factor. The reason that receptor is dangerous is that it causes cells to divide. So you don't want this left on continuously stimulating a cell to divide, or else you get a tumor, right? So an LDL receptor is a good example, and all growth factors are a good example of receptors that you want to use them just a couple of times and then destroy them. So typically what you'll do is pull them off the cell surface and send that receptor to the lysosome along with its cargo. In this case, the cargo would be epidermal growth factor because it's sort of a protein hormone-like substance, okay? And so a good example of another dangerous receptor is the opiate receptor in your nervous system, responding to endorphins after you exercise. That's why you feel good. It binds to the opiate receptor. But that needs to be a very highly selective process. You don't want to leave the opiate receptor displayed, otherwise you get addicted, okay? That's part of the problem. And so opiate receptor is another good example of a receptor that gets destroyed. It gets sent to the lysosome. And we can actually see that happening in this little experiment. And what I need to do is turn the lights down for you. Okay, so in this experiment, we labeled the 2 different types of receptors. We labeled the transferrin receptor that's on this cell, okay, with a red fluorochrome. Okay, so red represents the transferrin receptor that's going to get recycled. Okay, the opiate receptor is also expressed by this probably nerve cell, and it's labeled green. Okay, so when it's coming into the cell, what you see is both of them must be in the same compartment, right? Because everything is endocytosed off the cell membrane using a clathrin-coated mechanism first goes to the endosome, right? And so the early endosome is going to be yellow because red is superimposed on green. That makes yellow. So, and the reason that is you have red receptors, red transferrin, and green opiates are in the same compartment. So all these yellow-looking compartments are all early endosome. But that's where they diverge. What's going to happen to the transferrin receptor? It's going to go to the recycling endosome, and the ones that are just red, those are the recycling endosome, because it's retrieving the transferrin receptor and putting it back on the cell membrane. Okay. Now, what happens, the late endosome is where the opiate receptors go. They are not recycled, they're sent to the late endosome, because they're going to go where next? To the lysosome. Okay, so just get that down. If it goes to the late endosome, it goes to the lysosome. So you can tell where these things are diverging in the same cell based on the compartment that they occupy. Okay? All right, and a good exercise for you is based on that pH description that I went through for the LDL particle. How is it that you take in, iron is bound to transferrin, and the transferrin is bound to the transferrin receptor. So for homework, you figure out how you're going to uncouple the iron from the transferrin and the receptor and not destroy all the good parts, which would, you don't want to destroy transferrin, you want to reuse it. You don't want to destroy the transferrin receptor, you want to recycle it. How can you just get the iron off and let the cell have the iron without destroying everything at once? You have to play with the pH, okay? So that's a nice problem. Okay. Now, what's another pathway? Okay, so you got 3 pathways. You can recycle the receptor, you can send it to the lysosome for destruction, or you can actually send it across the cell. That's called transcytosis. And there's only certain types of cells that can do this. These are polarized epithelial cells. That doesn't mean they're electrically polarized, which they are. They're functionally polarized. So an intestinal cell has a luminal cell membrane, right? And then it's got a tight junction that prevents these proteins from diffusing into the basolateral membrane. So this membrane is very different from that membrane. Okay? So what you can actually do, and this is a good practical example, is so in a newborn that's nursing, right? It's getting a lot of its antibodies from the mother's milk. Okay, so that's going into the baby's GI tract, and it's not that the neonate is not producing high levels of acid, so it doesn't get really destroyed very well. And that child is actually producing what's called an FC receptor. That's going to recognize the mom's antibody, the FC portion of that antibody, and bind onto that Y-shaped immunoglobulin, right? So you can barely see that sort of green-colored antibody. And so you don't want to destroy that, so the last thing you want to do is take the mom's antibody and then send it into the late endosome, right? What you do is you send it first, of course, to the early endosome, but you don't want this to uncouple at low pH. You want it to stay bound, okay? And so it's going to go, and it's going to take a transport vesicle into the recycling endosome. And instead of going to the plasma membrane, it's actually going to go to the opposite membrane. This thing can recycle it across the cell membrane to the basolateral membrane, and at the pH that occurs in the bloodstream, it does dissociate because it's slightly different from the intestinal pH. And so this immunoglobulin is now delivered into the baby's blood supply and causes passive immunity, right? So there's reasons that you want to do transcytosis. There's reasons you want to recycle, and those are 3 different pathways that different cells have. Okay, so any questions about what's going on here? Okay, so let's distinguish phagocytosis real briefly from endocytosis. Phagocytosis at the cell membrane is very distinguishable because it's grabbing onto big cargoes like this cell, I think, these are like red blood cells that have been damaged, and so this cell is taking that up. And the way it does that is quite different. Instead of invaginating a clathrin-coated vesicle, it's using an entirely different process. It's using a process that's driven by the actin cytoskeleton. Now, when we get to the cytoskeletal section, you'll understand that this is a specific type of cytoskeleton known as microfilaments. Okay? And what happens is those microfilaments are induced to polymerize, and they form these big pseudopods. So instead of invaginating, you're actually sending these pseudopod arms and encircling that whole cargo, which in this case is a bacterium. Okay? And this little phagosome has a single lipid bilayer around it as opposed to the autophagosome. Okay, remember that. And so this polymerizes, and what's changing the polymerization state is actually the phosphorylation state of the membrane. So it knows what's going on based on the phosphorylation state. Okay, and then this phagosome is going to fuse with what? We want to destroy it, so where do we send it? So it doesn't go to the early endosome. That's for vesicles. This is a phagosome. It's huge. It's going to go to the late endosome, right, and then it goes to the lysosome. Good, so you got that. Okay, that just shows that. Okay. The last thing we really need to do is talk briefly about these 2 pathways. And this is the constitutive pathway, and I don't think I have anything specific. Yeah, okay. So Constitutive pathway is going to be, there's no targeting information left on the protein. It gets taken up into a vesicle, and we're going to just use sort of oversimplification. We're going to say all constitutive traffic, like collagen and mucus that a cell is producing all the time, is going to be butted off from the trans-Golgi network with COP1 coding. Okay? Remember, we use COP1 for a couple of different purposes. One is retrograde traffic from the Golgi, the cis-Golgi network, back to the ER. And under the cisternal maturation model, any kind of retrograde vesicular traffic between the cisterna are all COP1. Okay? The third thing we're going to use COP1 is for constitutive traffic directly to the plasma membrane. You don't go to the early endosome, you don't go to the late endosome, you go straight to the plasma membrane. And so if that cargo, like the collagen molecule, has no information about where to take it, it must be, it's not diverted, it goes straight to the plasma membrane. Okay? Now, so that's constitutive traffic. The third pathway, so we've talked about lysosomal traffic, constitutive traffic, is the secretory vesicle pathway. Okay, so this is what we call it as the regulated secretory pathway. And that's where we're taking something, usually a hormone is a good example, like insulin. And you package it into a very special type of compartment, and then you pull it, you butt it off the trans-Golgi network, and then it stays in the cytoplasm. It's actually in a special compartment, a storage compartment, okay? And it waits for a regulated signal to tell it when to release insulin, okay? And so what would that signal be? It would be a rise in blood glucose, right? So if you have something like a hormone or a GLUT4 that's stationary, you want to store it, and you don't want to display it until you get the signal, you use this pathway. Okay? And what's very specific about this pathway is it's a little confusing in this diagram because what it looks like, green is clathrin. It looks like, oh, it sort of fuses off, it buds off into a clathrin-coated vesicle. No, it does not. Okay? What it buds off into is this little compartment is actually pinched off by a process that's very similar to phagocytosis. So this pinching right here is driven by the actin cytoskeleton, and it is not clathrin mediated. So what happens is, in order to create this regulated secretory, it's actually called an immature secretory granule, is number one, the cargo has to self-aggregate. So insulin aggregates with the help of a couple of proteins. I've got them listed here on the outline. Chromogranin B and secretogranin 2 are different proteins, and what they're doing is they're getting all the insulin to self-sequester. So that's kin selection. I talked about that briefly. So you have something that self-aggregates into a particular pocket of the trans-Golgi network, and then the actin cytoskeleton basically just pinches it off. So a pseudopod comes around this side, and the pseudopod comes over here and squeezes off this big, huge particle. Now, this has got lots of stuff that you want to get rid of. You want to get rid of all the components and just concentrate the insulin into a highly compact insulin crystal, basically. I'll show you what it will. So what you do is you create this is pinched off, so now you have the immature secretory granule, and so you leave the insulin behind and you take everything else, all the rest of the mass of that immature secretory vesicle, and you butt off that stuff with clathrin and send it back to the trans-Golgi network. So you recycle it with clathrin, you don't butt it off with clathrin. So that's the distinction. You use actin to produce this, and then you take off all the excess so that you have a nice concentrated cargo of insulin stored in this mature secretory granule. Okay? So it's a little different than normal. But the way you get rid of all the extra volume and goodies is to use clathrin-coated vesicle and send it back to the trans-Golgi network. Okay. Questions about that? Now, once you've done that, the hallmark of regulated secretion is that this mature secretory vesicle does not fuse. It sits there. and it waits for some sort of regulated signal, okay? Now, so what's going to happen is it's going to wait for that signal, and then it's going to dock and fuse with the plasma membrane, releasing, in this example, would release insulin outside of the cell. So the signal that it's waiting for is a rise in blood glucose, and we won't go into that signaling mechanism at this point. So here's a nice picture, an EM of an insulin crystal that's actually being released in response to the beta cell detecting a rise in blood glucose. One of the things that happens in regard to insulin, remember that insulin is this long protein, and it's synthesized as a precursor. And the reason it is so that it forms the correct positions of all these disulfide bridges. Insulin has 3 different disulfide bridges. But this is immature. It's not ready to be secreted in the blood. When do you activate insulin so it's ready to bind to its target tissues like fat and muscle? Well, what happens is you have these prohormone convertases, which are basically proteases, and they're going to clip here, and they're going to clip here, and this little piece comes off, and because it looks like a little C, they call it C-peptide. Where does this happen? It starts to happen in the trans-Golgi network. So when you're selectively aggregating insulin together, that's when these prohormone convertases start to snip into the insulin. And so the modification starts there and it finishes in the immature secretory vesicle. So by the time this is a mature vesicle, it's got a completely active form of insulin. What else is inside that vesicle? C-peptide. And it turns out that C-peptide also has its own effects. In fact, it's pretty far along in clinical trials. They're going to start giving people C-peptide to mimic the secretory products that are actually released in the bloodstream when a normal person secretes insulin. So people will be, it turns out that the C-peptide appears to inhibit the inflammation that goes along with insulin's effect on the cardiovascular system. So it should have major effects on preventing cardiovascular events. Okay. Questions about this? So this is just, these are enzymes that are working inside the secretory vesicle. Okay. And the last thing I'll look at is, we've already talked about, this is like GLUT4, so this is at the target tissue. And so insulin wanders in here, activates the receptor. What we're going to talk about next time is we're going to start the signal transduction. And so we're going to find out exactly how does this signal get to the regulated secretory vesicle. So this is a GLUT4 is stored in a regulated secretory vesicle, and somehow it tells it to fuse with the plasma membrane. So we'll go into that. Okay. I think that's everything. Yeah. So that finishes quiz-free material. Oh, okay. Oh, don't worry. I had to be up there.
Okay. What ended up happening was I didn't realize I had sent an email to my HSC account. So
Okay.
I was back there. But
Let's see.
Do you have the first lecture recorded? Like after the quiz?
Yeah.
You do?
I put it on YouTube.
You put it on YouTube? Okay, I know.
Oh, okay. Thank you.
You have anything?
I have.
Well, I don't have class on Tuesdays and Wednesdays.
Did you have any vote?